Hello everyone. Today we are going to discuss a topic that is experimental unit. Why the experimental unit is important? Because most of the time researchers think that experimental unit is always animal and it will directly affect your results and conclusions. Through the definition, so the experimental unit is a smallest unit that receive a treatment individually. Remember this line, any unit that receive the treatment individually is experimental unit. Not always a rat, not always a mice. If you are doing a experiment with the animals, so any unit that receive the treatment independently is known as experimental unit. Why we go with the wrong approach? Because we think that every time the experimental unit should be a mice or a rat or any animal. Yes, most of the time this is rat or a mice or an animal, but not every time. So the rat can be a experimental unit if we provide the treatment directly to the rat. Means if there are four rats in a cage and we are giving the intraperitoneal injection to individual rat, in that cases the rat is experimental unit. But in sometimes cage could be a experimental unit if we provide the treatment not individually to the rat, but in the form of diet or sometimes drinking water. The treatment is mixed in drinking water. In that cases, the experimental unit is different. We'll take the cage as a experimental unit, not the rat as a experimental unit. Sometimes, if we are doing some experiment with the pregnant female and we have to find out the teratogenicity like effects that could be find out in the pups that are delivered by the female rats. So what we think that we have to calculate the effects from every pups and should be involved in our calculations of the sample size, but that is not correct. In that moment, if we are taking the calculations or the findings from the pregnant female and female are taking their diet in a litter and after some time it gives birth to suppose six or seven pups, so at that time we will take the litter as the experimental unit, not the pups as the experimental units. Sometimes we think that the rat is always a single experimental unit, but that is not correct. If we are going to find the effect of some drug or the some interventional agent locally, suppose we have to find out the effect of any NSAID drugs locally, and if we are providing or we are intervene that drug in the right and left thigh, means in the right thigh we are introducing the drug A, and in the left thigh we are introducing the drug B. So at that time there are they calculate it as a different units for the same rat. We are not taking as a single unit. That will be incorrect method to include in our studies. Sometimes there are different or multiple experimental unit could be present in a single experiment at different time periods. Like if you are giving the drug A in 1 week, leave the experiment for the washout period, like in the second weeks or for sometimes 15 days. And we are now introducing the drug B, another drug for the next week. So for the first week, the rat will be calculated as a different unit, and for the third week, the same rat will be calculated as a different experimental unit. And sometimes the scenarios is different. In a single experiment we have to calculate the different units like the split plot experiments, in which different treatments are applied at different level, and we find more than one experimental unit within the same experiment. Let's take example. Suppose there are four cages, and in each cage, five rats are there, and we are going to find out the two different observation. One observation for any drug that is introducing in the animal individually. Suppose drug A we are going to introduce individually to the rats, and another finding we have to calculate some activity like behavioral activity depend on the temperature of the room. So one unit or one variable we can call as the drug. In that case, how many units we have to calculate? There are 20 units because there are four cage, and in each cage rats are present. So then total units we have to calculate as 20 units. While when we are calculating the effect of the temperature, so here we can see there are four cages at different places. So every cage taken the intervention from the different place. We should calculate as 4 units for the temperature. Why this experimental unit is important? Here we can see like in scenario one, treatment is given individually to every rat. So total units here we can calculate as 24 because there are 24 rats in the experiment. So sample size we can calculate small n is equal to 24. In another scenario, like you can see in the scenario 2, here we can see there are four cages and the treatment is provided through the diet, and in each case there are six rats, but the number of cages is four. In scenario 1, where we can see the introduction of the treatment or the intervention to the individual rat, so the sample size is 24, while in the second scenario where the treatment is given in through the diet, the sample size is 4. So that is the difference. Second is that ensures the treatment are applied independently. Suppose there are six rats in one cage, or you give the treatment each and every rat independently. So we can find out the valid comparison or valid result. While suppose in the other scenario there are six rats and you are giving the treatment only one rat, and you are going to take the observation from all the six rat that are present in the cage, that in that case you will not find the correct findings and you will get the invalid comparisons or the misleading results. Third is it helps choose the correct statistical test. How suppose there are two scenarios. One we give or we intervene the animals individually, and what we will find the outcomes slightly different from each other. While in the second scenario, suppose you give the treatment through the diet, that in that case that is quite possible that you find the outcomes more precise, more equal to each other, that will change your result at the time of application of the statistical test. So in that case, in the second case, you may find the fake precisions or wrong statistical outcomes, and it is called as the pseudo replication. Of course, it will improve the reliability of the results. Yes, it allows the valid conclusion to be drawn. So the possibility is that the each rat uh, gives the more similar results, and you will got the more precise. So your conclusion will be overestimated, and that will lead to uh, disturbance in your results and your research also. Let's take some MCQ examples so we can better understand the finding of the experimental unit. Question one is, a researcher is investigating the effect of a new anti-inflammatory drug in rats. The study consists of three treatment groups, a control group, a low dose drug group, and a high dose drug group. Each treatment group contains four cages, and each cage houses four rats. The drug is administered individually to each rat by intraperitoneal injection. At the end of the study, inflammatory markers are measured in all rats. So, which of the following is the experimental unit in this study? The most important line in this case is that the drug is administered individually to each rat by intraperitoneal injection. Each rat means here the rat is treated individually. So, the correct answer will be the correct answer is the individual rat. Next, a researcher is studying the effect of a special diet on weight gain in rats. A total of 24 rats are housed in 4 cages with 6 rats in each cage. The diet is applied. The diet is applied at the cage level, meaning that all rats within the same cage receive the same diet. The cages are randomly assigned to treatment conditions. How many experimental units are there in this study? So the main line here is the diet is applied at the cage level, meaning that all the rats within the same cage receives the same diet. So the correct answer here should be cages, not the rats, but cages. So how many cages are there? 4 cages. So there are 4 experimental units. Next question is, a researcher wants to evaluate the effect of a new water treatment on fish growth. A total of 24 fish are housed in 4 tanks with 6 fish in each tank. 2 tanks receive the new water treatment while the other 2 tanks receive normal water. The treatment is applied to the entire tank, so all fish within a tank receive the same water condition. So, how many experimental units are there in this study? So, the main line here is the treatment is applied to the entire tank, not to the fish. So, here the experimental unit must be tank, not the fish. So, how many tanks are there? There are 4 tanks, and in this case, the correct answer is C. 4 tanks as an experimental unit. Fourth question is, a researcher is investigating the effect of drug administered to pregnant rats on the birth weight of their offspring. The drug is given to 8 pregnant female rats. After delivery, each dam produces a litter of approximately 6 pups. Birth weights are measured for all pups, and the average birth weight of each litter is used for analysis. So, how many experimental units are there in this study? So, the important thing is here we are going to do a study on the pregnant female rats. So, always the treatment is providing through the litters. In that cases, the litters are always experimental units, not the pups, not the dams, but the litters. We have to calculate the litter as a single unit, a single experimental unit, because we are going to provide the diet through the liters. So these cases are for easily finding out the experimental units for our experiments. Correct answer is C. 8 liters will be count as the 8 experimental units. How we can adjust the multiple measures from the same experimental units, because suppose in the case of the pregnant female rats, there are 12 pups. We cannot find out the individual observation from all 12 pups. What we can do, we can do a average of their readings, and we can put that average of that readings in our experimental calculations. And we can also do a repeated measures ANOVA when the measurements are collected different time interval. And we can use also mixed effect model for repeated or hierarchical data. What are the nuisance factors? Some factors that can also alter your results rather than drugs and some other interventions. Drugs and intervention that are also known as the factors of interest. But a nuisance factor, like suppose we are going to do a research on the weight of the rats, and we are introducing some drug to the rats. So drug is the factor of interest that may change or may not change the weight of the rats. But some other factors also, like stress, like the position of the cage, that causing or that making the stressful condition for the rats, that factors also can change the weight of the rat, because that may lead to decrease in the weight because of the stress. So that factors are known as the nuisance factor. So we can remove that factors by some methods like the randomization. If we do the proper randomization of the rats, the nuisance factor can be removed from, or can be balanced in that conditions, like the blocking factors going to divide the animals in two groups. So it is quite possible that the more females are present or present in the group 1, and the less females are present in the group 2, that will affect your results. And we have to also instantize the conditions like the housing, diet, temperature, lighting, and the handling procedures, that will also affect your results. And that balancing of the nuisance factor is also important. If that is not possible to complete removal of the nuisance factor in every experiment, so that is very important to balance these nuisance factors, and that will lead to the good statistical results. So in this lecture, the most important thing is experimental unit is not always a rat. So point to remember in this lecture, that the experimental unit is not always a rat. It is equal to the sample size, and experimental unit is always calculated depend upon the intervention that is given to individual unit, not to the individual rat. Thank you so much. I hope this lecture will be beneficial for your experiments. Thank you so much.